Hello students. Today we are going to learn about mitochondria, which is considered as a powerhouse of a cell. Mitochondria is a plural word. Its singular is a mitochondrion. Okay, unless specifically stained, we cannot observe it. Okay, we have to specifically stain it to observe and are not easily visible under the microscope. So without any staining procedure, any specific staining procedure, you cannot observe under the microscope. So it requires special staining. The number of mitochondria per cell is a variable, means cell to cell it will be different depending on the physiological activity of the cell. Means how long the physiological activity is done by the cell and how much energy is required by the cell. According to that, a number of mitochondria will be present in that particular. In terms of shape and size also, considerable degree of variability is observed. Means size and shape also varied from one cell to another, organism to organism. Typically, it is a sausage shape or a cylindrical having a diameter of 0.2 to 1.0 micrometer, and average it will be 0.5 micrometer, and length will be 1.0 to 4.1 micrometer. If you observe any mitochondria, each mitochondria having a double membrane bound structure. Okay, so each mitochondrion is a double membrane bound structure with the outer membrane. Here you can see this is the outer membrane. Okay, so outer membrane and inner membrane, two membrane will be there because it is double membrane bound structure, and dividing its lumen distinctively into two aqueous compartment. Because of the double membrane structure, it will divide into two structures, two compartment, and that is outer compartment and the inner compartment. Innermost compartment, which is here you can see bluish color, okay, dark bluish color, that is a matrix. So innermost compartment is called matrix. The outer membrane forms the continuous limiting boundary of the organelle. Here you can see in the diagram. Okay, so this is the organelle, that is a mitochondria. Here it is the limiting boundary, outer boundary. Okay, and that is a continuous. The inner membrane, if you look at the inner membrane, two boundaries are there, inner membrane and outer membrane, out of which outer membrane is a continuous. Okay, whereas inner membrane forms the infolding. Here you can see this is the inner membrane. This outer membrane is continuous, but inner membrane folds, okay, like this. Okay, it will be like this. Okay, see here, this is the structure, that is the inner membrane infolding. Okay, and that infolding is called cristae. This infolding, that is the inner membrane infolding, is called cristae. Singularly, it may be called as a crista towards the matrix. Here you can see towards the matrix infolding is generated, and that is called crista. Now let me try to understand the structure of a mitochondria by using this diagram. So here you can see this is the outer boundary of a mitochondria. Okay, this is all structure of a mitochondria, and this is the, you can see here, this is the outer boundary, and that is called outer membrane, and that is a continuous membrane. Okay, and it will contain the transport proteins for the shuttling of the pyruvate into the mitochondria. We'll learn what is the shuttling of a pyruvate into mitochondria. One pyruvate molecule will be transported across the membrane of this outer membrane, and that's why it is called shuttling pyruvate. Okay, means shuttling of the pyruvate may occur into the mitochondria. From cytoplasm or a cytosol, this pyruvate will be transported across the outer membrane, and that's why this phenomena is called shuttling pyruvate. Okay, so we will learn in detail in next lectures. Okay, so what is the outer membrane function? It contains transport protein. You know very well now what is the transport protein. Okay, for the shuttling of a pyruvate into mitochondria. Now, if you go inside, you can see here, this is the second membrane, that is a whitish color. Okay, the second membrane is not continuous. It is having an infoldation. Here you can see this is the infoldation, and that infolds are called crista. Okay, it is highly folded and so as to increase the surface area. Okay, if it will require for the increasing the surface area volume ratio. So surface area and volume ratio will be increased by crystal. Okay, and this is the membrane, that is a whitish color membrane, is called inner membrane. Okay, and it will be contain several enzymes and several chains and several proteins which are importantly utilized during electron transport chain. and also it is responsible for ATP synthesis, and that's why it will having one enzyme, very important enzyme, which is participate during ATP synthesis, and that is ATP synthase. Okay, and that is for the oxidative phosphorylation, and this is the phenomena by using which ATP generation occur. And here this main function of this mitochondria is to generate or synthesize ATP, and that's why we call it as a powerhouse of a cell. Okay, because ATP is a sole source of energy. So energy will be produced or synthesized inside the mitochondria, and that's why we are calling it as a powerhouse of a cell. Then here you can see in between two membranes. This is the outer membrane, this is white border you can see here, this is continue border, white border, and inner border which is discontinue, and it is having a folds. So in between whatever the pinkish region is written, you can see here, okay, whatever the pinkish region is there, and that is called intermembrane space. Okay, that is a small space in between inner membrane and outer membrane, and it will be required for the accumulation of the protons. So during the oxidative phosphorylation, accumulation of proton will occur in this space. Okay, in between inner membrane and outer membrane. So it is the small space to quickly accumulate the protons. Okay, and the yellowish color, whatever you can observe here, okay, yellowish color region, and that is the innermost region, and that is called matrix. Okay, and has appropriate enzymes. So many enzymes will be there inside that. Okay, and it is very important structure, and very important functions also carried out inside that. So has appropriate enzymes and a suitable pH for the Krebs cycle. We will learn what is a Krebs cycle. Okay, that is also called TCA cycle, but we will learn in next lectures. So matrix, then outer membrane, we can start from the outer membrane. Okay, then inner, then you can see crista. Okay, this will be the crista. This is infoldation is the crista. Then inner membrane. In between outer membrane and inner membrane, whatever the pinkish color, light pink color region is up here is called intermembrane space, and the yellowish region is a matrix. As we had discussed, the crista is very important to increase the surface area to volume ratio. Okay, and the second very important function is two membrane have their own specific enzymes. We have talked about the membranes. Okay, that is outer membrane and the inner membrane. Both membrane having its own specific enzymes associated with the mitochondrial function. So let me see what are the enzymes. Here you can see this is the mitochondria, and we have already discussed about the structure. This is the outer membrane, inner membrane. Inner membrane is a folded. Okay, and that is called crista. Then outer membrane is there, inner membrane is there. Their own DNA is also there. We will discuss what is their own DNA of a mitochondria, but here we are discussing about the enzyme present in the mitochondria. So here you can see, suppose if you look at this point, okay, this is the membrane, that is the inner membrane. Okay, and this is the outer membrane. So here you can see in the outer membrane, enzyme of a lipid metabolism is present. Okay, here you can see it is written, see here, enzymes of a lipid metabolism is present on the outer membrane, and along with that some porins are there. Porins are the pathway or a ways for some molecule to enter or come outside. Okay, so that is called porins, and inside that you see here, this is the outer membrane and this is the inner membrane. In between these two, you can you know very well, that is the intermembrane space. Okay, here intermembrane space. Inside the intermembrane space, you can see a nucleotide kinases are present. That is the one type of enzymes. Okay, that is a nucleotide kinases, and here creatine kinase is also present. So these are the enzymes which are present in the intermembrane space. Okay, here in the inner membrane, you can see here some enzymes are embedded in the membrane. So one of the most important enzyme out of the inner membrane space, inner membrane, on the inner membrane incorporated, and that is ATP synthase. Okay, this is the most important enzyme because it participate in the ATP synthesis. Okay, that's why it is very important. ATP synthase enzyme. Then here you can see transporters are there. Okay, this membrane is exactly similar as we have learned already, a cell membrane. Okay, that's why some proteins are incorporated or membrane bound proteins are found in that. Okay, and so here you can see some proteins will act as a transporters. Okay, some enzymes of oxidative metabolism is also present here. You can see respiratory chain related enzymes are also present over here. Okay, we will discuss in detail. Okay, so this is all about the enzymes, and even inside the matrix also, in matrix space also, so several more enzymes are also present. So we will discuss in later on. Okay, in next chapters. Mitochondria are the sites of the aerobic respiration, and they produce cellular energy in the form of ATP, and hence they are called as the powerhouses of the cell. Now let me try to understand more with this diagram. So here you can see this is the cell. Okay, inside that there is a mitochondria, and surrounding that there is a cytosol. So surrounding the mitochondria there is a cytosol. Okay, and this cytosol will have some more enzymes which are responsible for the glycolysis. Okay, so whatever glucose is entered in the cell, that will be degraded out or break down by using glycolysis in form of a pyruvate. And finally this pyruvate from the cytosol, which is formed inside the cytosol by using glycolysis process, okay, this pyruvate will be shifted to the mitochondria, means they will now enter, means pyruvate whatever are generated will be enter inside the mitochondria, and they will enter into citric acid cycle. So citric acid cycle is also responsible for the ATP formation. Okay, and the products of the citric acid cycle will further go, means the product which are including NADH2 or FADH2, they will be undergo oxidative phosphorylation, and by this way it is also responsible for the production of ATP. So by this way you can say that inside the cytosol there is a glycolysis process by using which ATP can be formed. Inside the mitochondria, when pyruvate enter, at that time it will enter in the citric acid cycle, and that is responsible for ATP formation inside the matrix. Okay, this process will occur inside the matrix of the mitochondria, whereas these process, the products of the NADH2 and FADH2, means products of the citric acid, which is the NADH2 and FADH2, will be further proceeded inside the inner membrane. So in the inner membrane, oxidative phosphorylation will occur. Okay, and that is also a pathway for the ATP formation. So by this way ATP will be formed. So most of the ATP or energy related resources will be formed inside the mitochondria, and that's why we can say that a mitochondria is the powerhouse of the cell. Along with the enzyme for the citric acid cycle or a TCA cycle, matrix also possesses single circular DNA molecule, and that is its own individual DNA molecule, that personal DNA molecule. Okay, and a few RNA molecules are also present in the matrix. Then ribosome, that is a 70S ribosome. See, it is very interesting to note here. Ribosomes are having 70S, means mitochondrial ribosome which are present in the mitochondria having 70S ribosomes, and the component required for the synthesis of a protein. So inside the matrix there are several other components also. Here you can see this is the matrix granule, ribosome, mitochondrial DNA, their own DNA. Okay, so it will be replicate their self, means this mitochondria will be replicate their self, don't require nucleus related messages. Then ATP synthesis, here you can see this is a pinkish color. This pinkish color region is ATP synthesis. Okay, and this pinkish color region can be divided into two groups, F1 and F0. So F0 will be embedded inside the membrane, that is the inner membrane, and outer side here you can see pinpoint like structure, and that is a F1 granule, and so this is called F1 granule. Okay, this is a whole part of ATP synthesis, and then you can see intercrystal space, peripheral space. Okay, then porins. Okay, and then here you can see inner membrane boundary, crystal membrane matrix, crista, which already we have discussed. So it is very interesting to note here that if you compare a prokaryotic cell and eukaryotic cell, then you can say this prokaryotic cell having these structures, okay, eukaryotic cell having these structures, and both having a common structures like cell membrane contain DNA, ribosomes and cytoplasm. But ribosome is having in both the cells. Okay, it is present in both the cells, but is several difference. With several difference, you can see here in the prokaryotic the ribosomes will be found will be of 70S. Okay, whereas in the case of a eukaryote, if you see, the eukaryotic cell will have a ribosome of 80S. But here you must note a mitochondria which is present in the eukaryotic cell, that is an organelle. Okay, inside that whatever the ribosomes are found will be of 70S. and that is as per similar as the prokaryotic cell. So it is here you have to note down that whole cell, okay, the eukaryotic cell will have 80S in their cytoplasm, but inside the mitochondria they will have exactly same as the prokaryotic cell ribosomes. As we have discussed, mitochondria have its own DNA, own ribosomes, and that's why they can replicate their self, means they can divide by their self, and that method is called fission. Okay, so here you can see this is the mitochondria. Suppose it will elongate first. Okay, it will increase in length first, that is elongated mitochondria. Then afterward one process is called fission. Here you can see some molecule will be participate here. Okay, and as a result of that, a fragment of the mitochondria will occur. Here you can see by using some proteins. Okay, and by this way it will be divided into two small fragments, and they will act as an individual mitochondria. So by this way, this is the process of fission. Okay, and by using this process, mitochondria will be divided into two, and we can say replicate into two. So this is your homework for today's lecture. And before we finish this lecture, let me summarize all topic by using one animation video. So look at this video.
Called the powerhouse of the cell, mitochondria are sites of aerobic respiration and produce cellular energy in the form of ATP. The number of mitochondria in a cell varies depending on the cell's metabolism. Muscle cells, for instance, need a continuous supply of energy, and hence they contain a large number of mitochondria. Microscopic studies have revealed that a typical mitochondrion, which divides by fission, is sausage-shaped or cylindrical, with a diameter of 0.2 to 1 microns and a length of 1 to 4.1 microns. It is a double membrane bound structure consisting of an outer membrane and an inner membrane, and each membrane has its own specific enzymes associated with the mitochondrial function. The membranes divide the mitochondrial lumen into two aqueous compartments, the outer compartment and the inner compartment called the matrix, that contains a single circular DNA molecule, a few RNA molecules, 70S ribosomes, and the components required for the synthesis of proteins. While the outer membrane forms a continuous limiting boundary, the inner membrane folds towards the matrix to form infoldings called cristae. These cristae increase the surface area of the inner membrane of the mitochondria.
In next lecture, we will learn two topics, and that is plastids and ribosomes. So till that, goodbye.